The next application of biotechnology is to create genetically engineered insulin. Now, insulin is a hormone that is secreted by pancreatic gland. Pancreas secretes insulin. This insulin, it helps our body to take up glucose. So whatever body cells are there, if our body cells take up the glucose, only then our body will get energy. So for the process of uptake of glucose and convert it into energy, insulin is required. Now, due to some defects in the pancreas, for some people, pancreas is unable to make insulin. So there will be insulin deficiency in the body. That is why insulin in the form of tablets or injections have to be taken by those patients. And the disease is diabetes. Initially, insulin was taken from slaughtered cows and pigs. The problem, if the insulin was taken from cows and pigs, was that it causes allergy to humans. That is why it was very necessary that insulin hormone has to be genetically human itself. For that, what they did is they isolated the gene that produces this hormone and they started culturing it in bacteria like E. coli. So as the bacteria asexually reproduces, the insulin gene will also be multiplied and that will lead to the production of insulin hormones in the culture medium. But there was still a challenge because these hormones, what was produced, was in immature form. And that needs to be converted to mature form. So immature form of insulin means insulin is made up of peptide chains like A peptide, and then B peptide, and both these chains are binded with a peptide which is called C peptide. This is immature form of insulin hormone. Now, when it is produced in our body has got mechanisms to remove this C peptide and thus making it into mature form by adding disulfide bridges here. So this was a challenge that how to make the immature form of the cultivated uh, this was a challenge that how to make this immature form of insulin into mature form. Now, Eli Lilly and company solved that problem. Eli Lilly is an American company who solved this problem. What they did is they cultured gene that codes for A peptide and the gene that codes for B peptide. Separate, there will be E. coli strains that has gene that codes for A peptide. There will also be a culture of E. coli or bacteria that contains the gene that codes for B peptide. After isolating them separately, A peptide chains as well as the B peptide chains. And now it is ready to be marketed.